Some brief announcements. First is that Cycle 6 will run from October 15th to October 23rd. It'll be in the span of 1.5 weeks. So 15th to the 23rd will be Cycle 6, and then Cycle 7 onwards. No Cycle 5 quiz due this Sunday. Do whatever you have to do this long weekend. Rest, enjoy, catch up, whatever you have to do. It's your weekend. There's no quiz due the weekend of the midterm, which is the weekend after. So no quiz this weekend and no quiz the weekend after. The next quiz is due October 26th at 11 p.m., and that will cover Cycle 6. Dr. McDougall-Shackleton and I will be holding a midterm Q&A on Thursday, October 16th from 8 a.m. to 9:20 in this room, NCB 101. The questions pertaining to cycles 2 to 5, I will be there from 8 to 8:50, and then Dr. McDougall-Shackleton will take over from 8:50 to 8:20, 8:50 to 9:20, and she will transition directly into her lectures. So if you're in this class, then you're welcome to stay and go on with her lecture. The biology mentorship group is holding review session and mock midterm next week as well, so please go to the BMG tab for details. There's lots of help next week and this week. I'm also hosting my office hours this week and next week, as you know. Midterm exam announcement will be sent out soon. Make sure that you read the announcement, and all that information will be there for you in one document. So this is my last lecture to you this term. But I do teach 1002B next term, but I teach the second half. Dr. Dennis Maxwell will teach the first half, so I won't see you in this setting until mid to late February of next year. So happy everything that comes from now to then. I posted this supplementary resource tab for you, and it gives you a lot of information on Success Beyond the course. I would highly recommend, if you have time, to just go and have a look at this. I know that a lot of this information is out there: how to get a research position, how to find a good mentor, how to build a CV, how to give a good, impactful interview. I tried to put a lot of personal experience into this, so there was no gatekeeping here. This was just, take advantage of all the resources that are out there. Your softs are doing a lot of this stuff. Your mentors will help you out with this. Go to as many as you can, but I have this there for you. When I used to teach 3rd and 4th year genetics, I found it was really small class sizes, and I was just able to communicate a lot of this stuff a lot better. And with a large class, I find it very difficult to help you beyond the course, but this is the best I can do. The number one thing I find where students struggle is to start building their CV very early. Over the last 4 to 5 years, I've had the opportunities to sit on selection committees for a lot of things, and I learned a lot of things in that. I just want to share some of those things with you. I think it's important for you to start building your CV now, and make sure that whatever you put on your CV is very tangible. So if you just say, I gained experience in something, it's not enough. Well, we need to see that CV. That CV will help you get shortlisted. Shortlisted means to be called for an interview. We're not going to call for an interview, call you and go, hi, are you there? Can I see you? We're going to call you for no. It's just, this is your CV, this is your GPA, this is based on all of this, you'll be called for an interview. And then you can shine, right? But if you don't even get to the interview process, then it's not going to happen. When you get to the interview process, your competition level is different, because now everything in your CV, which is going to look stellar, everyone has that. So you have to now jump to the next level. So each time you get up there, your competition is changing, and you have to change what you say and how you handle that competition as you go up that ladder. So I talk about that. Do you know how many students I know that have so much experience and so many extracurricular activities, and they've done so much, and they don't know how to present it in the CV? It doesn't look impactful at all. I'm like, really? Like, this doesn't even look impactful. You should write it in a way like, I gained experience. What did you do with that experience? Give some outcomes, give tangible evidence. Well, I published something. Here's the publication. I taught someone this material. Here's who I taught. I did a workshop. Here's the workshop with the information that I had, what I learned. the experience I gained. I built something unique. I came up with ideas for this project. I didn't publish the paper, but those are my ideas from what I learned. These were the ideas that led to this publication. So tangible stuff that you can see, not just, I gained experience, good for you, right? Good for you for the experience. That's not gonna work in your favor. So please make sure that your CV is very, very tangible. It's full of tangible evidence. What's the outcome? Oh, you know what? The outcome is that I used that experience and we raised money. We did fundraising. This is how much we raised for this cause. Boom, tangible. Got it? So tangible, tangible, tangible. And just understand that, for the interview, a lot of people ask, like, what do I say? What do I do? It's simple. The formula is simple. Understand what they're looking for and deliver. That's it. Understand, what do they want? I will deliver that. So in the interview, do your research, talk to people that are in that field. What are some strengths that they're looking for in someone with this career? What do they want? Ultimately, guys, you know what they're looking for in the interview? Are you a leader or are you a follower? Do you have a good temperament? Are you someone that's going to be, you can get along with? So those are a lot of things that they're looking. They're not looking for you to be in that profession. Oh, I want to be a doctor. Look at me. I already am a doctor. I know everything. You can know nothing about the medical field. Are you the right person to study medicine? Okay? So please understand what they're looking for and deliver. So find a good mentor if you can. This is the time in your life, find a really good mentor, ideally someone that is already in the position that you want, and ask them, what did you do to get there? What are some pros and cons? What's the job like? What do I have to do to get there? Tell me all the times you fell and you had to get up, What I want to avoid those mistakes. What should I pack in my backpack and have all the tools to be ready? These are such good things that you can do to get there. Your education is important and all this, but I'm telling you guys, a lot of things that are not taught in undergrad are so much more important than just your GPA. Your GPA only gets you so far. This is what people think a success map looks like. They think it's just a linear trajectory. This is what it actually looks like. You all know that. You have to fall in order for you to get up and learn from those lessons and get right back on the horse and keep going. So anyone that tells you that their trajectory was like this, they're lying. Or they're not comfortable enough to tell you, or they have too much of an ego to tell you. This is what people's trajectories actually look like. So one thing I want to share that may come out very blunt, and this is just something that I don't think anyone will just maybe come out and say it to you, but here's the thing. I'm old enough to say this. First thing is, it's important right now that you choose the career that is absolutely meant for you. That is important. Don't choose the career because you're trying to make somebody else happy. You're trying to make somebody else proud. You are doing it because somebody else really wants this for you. You're not doing it because, well, it's lucrative. It makes a lot of money. And, my family did so much for me to bring me here, and I want to earn money. I want to give them all the things in life that we never had growing up. I want to do this for them. Don't do it for that reason. Don't do it because it's actually, you know what? If I study this field, there's a whole bunch of jobs out there. There's a whole bunch of them. I can get a job right away. Don't do it for that reason. And you might be saying, well, Nikki, those are pretty good reasons. But I haven't said the number one reason why you should choose the career you should choose. You should choose it based on your passion, what you truly enjoy. Because you know what? Also choose it on what you're really good at intrinsically. Because people cannot compete with your genome. They may want to compete with your genome. Be like, how come you're so good at this? How come they just know what to do? Because it's intrinsically in you. It's part of your genes. Genes don't lie. Genes are the ones that are like making you, right? So you just have an advantage. You were born with the advantage. So use that to your advantage and ask yourself, what makes me happy? What makes me internally happy? You have to be the one to set your alarm clock every single day, wake up in the morning for the next 40 years. You, not the person you're going to do this for. And you might get all the pleasures, you'll be like, oh my gosh, I did it. I became this. And you have the bells and whistles and parties and congratulations. It's going to be anticlimactic. You're going to be like, well, it's done now. Now you have to be able to make sure that you like it. You go to work. So it's not about liking your boss. I don't like my boss. I don't like the people I work with. You're going to encounter people you don't like, people you don't like to work with. Your boss is not going to be always a perfect boss you always wanted. But you know what will be perfect? Is your job, the stuff you're doing. You're passionate about it. You put up with other stuff. But if you already don't like what you're doing, it's going to be harder for you to put up with your peers and to put up with your colleagues and all the people you have to work with and your customers and your, et cetera. It's hard enough. Find that passion, guys. Don't go in it for the wrong reasons. Don't go 15 years down the road like I have seen people do and come back and go, I'm not happy. I feel empty inside. I'm not happy. I'm supposed to be happy. I'm smiling, but I'm not happy, right? And you project that onto your children. You project it onto your spouse. You project it onto your other family members and your friends. It's a reality, okay? And I'm sorry I sound like a mom. My apologies, but this is just, I could not sleep without letting you know all this information. Start now, start early. You can do it and go in it for the right reasons and be happy. This is your exam booklet, the front cover of what it will look like. You have to make sure that you write the full name as it appears at the registrar's office. If you have a nickname, don't write that nickname here unless it is appearing in the registrar's office. So we take the list from the registrar, match it up, and that's how these papers are matched. Your username is everything that comes before the @uw.ca. This is your student number. The back of this page is blank, and the rest of them have the multiple choice questions. There are 40 of them, as you know from the midterm exam tab. This is version A. There are 4 versions in this exam, A, B, C, and D. When you'll be sitting such that different versions are writing around you. This is the sheet that will be graded. This booklet will not be graded, but you still have to hand it in. It won't be graded though. So you can do whatever you want in this. You can draw pictures of Mickey Mouse and Donald Duck. We don't care. This is what's going to be graded, your bubble sheet. This will be scanned and it'll go through a program and it will grade it. Guys, it is absolutely vital that you fill out and identify your bubble sheet. You have to write your full name as it appears in the registrar's office, your username, date, student number, and bubble in the correct version. Go back to your booklet. Your version is right there, so you're going to bubble in version A. If you bubble in the incorrect version, your bubble sheet will be graded using the incorrect answer key because that's not the version you wrote. That's going to reflect your grade. So please be mindful of that. Write clearly, like someone in grade 1 has to read it because the machine has to read it. Please write extra clearly. People get so nervous that they forget to identify their bubble sheet. They're like, where are the questions? Where are the questions? And I see this because I'm doing my rounds. I see it. Where are the questions? And they're doing everything and then they hand it in and they're like, oh, I forgot to write my name. I forgot to write this whole top part. Now imagine a class of 2,500 students, 3 students forgot to identify their bubble sheet. 3 students have it all blank. I look at all 3 of them. Do I know which one belongs to who? No. All 3 of them get a 0 on the midterm exam. I don't want to have to do that. Imagine I won't sleep at night. Imagine what's going to happen to you. So it's important that you fill that out. Can someone tell me why I have to give them a 0? You're going to be in a position like this someday, right? You're going to be in a position where you're talking, you're making decisions, you have to keep things fair, you have a job where you have responsibilities. So what would you do as an administrator or as a prof? Why would you say, no, you have to give them a 0? What could be the domino effect down the road? The domino effect could be they'll say, well, I'll write the makeup. I'm sorry, I forgot, I'll write the makeup. Would you be okay with that? If a group of students wrote the makeup exam after this incident, would that be fair? No. They've seen the exam already. They've seen the exam. They've done the exam once. They're going to do it again with minor little changes. They pretty much saw the whole exam. They'll get a much higher grade second time around. We'll look up all the answers. Go home and look up all the answers. They've seen the exam. So we can't do that. So you might say, okay, that's okay, Dr. Strand. Just don't make them write the midterm. Reweigh their final exam. Make them write a 68% final, right? Skip the whole midterm. Is that fair? I'm asking you guys, is that fair? You don't have to say no. If you think it's fair, nod your head, say it's fair. Do you foresee something in the future? What do you foresee in the future that could happen? If I said, no problem, you forgot, I'm so sorry, but next time remember, you can write the final exam. There's no midterm, I'll reweigh it. I'll push the weight of the final, midterm onto the final, you write a 68% final. Over the years, what's gonna happen? What could happen? Who could take advantage of that system and how? You could come to the exam, look at the exam, do the exam, and then decide the exam was too tough. I didn't like the questions. I don't think I did well. I'm going to, quote unquote, forget to identify my bubble sheet and hand it in. I forgot. Oh, you forgot? No problem. Here, you can write the final. You got to pick. You got to choose. I have to be fair for you. It's no skin off my back, guys. If 3 people come and just, say that, I'll go, fine, go write the final, whatever, sure. But you will come to me and say, that wasn't fair. I failed the midterm. I identified my sheet, so therefore I got penalized for it because I identified my sheet. I wrote my name. I bubbled everything incorrectly, and now you're penalizing me for it. I don't get to write the final exam and forget the midterm, but they do because they forgot to write so you see what I'm saying? It's not going to work like that. I have to keep things fair for you, okay? We don't want half the class doing this. I mean, it's a hot mess. So please, the bottom line is, please, please, please fill this out. This can have an impact on your grade. I'm warning you, please. Other thing, guys, this bubble has to be absolutely complete. A machine is reading this. It'll look for darkness. So if the correct answer is C, you circle C, nice and dark. If you go, oh, it's not C. Oh, sorry, I changed my mind. I double checked. The correct answer is actually A. If you circle A, make sure you erase the C. If it sees 2 or more bubbles, you get a 0 for that question. So make sure it's just 1 bubble. Second thing, if the erasing is not complete, it will see it as 2. Invest in a good eraser. Spend that extra buck, it's worth it. And erase really, really fully and completely so that there is no darkness. The darkness should not be detected by the program. As you know, we have alternate grading. We'll calculate your grade 2 different ways, standard and midterm, low, and whichever one is a higher mark, we'll give it to you. Do your best, please, not to write a 68% final. If you have to miss the makeup, it is what it is. It's life, right? Unforeseen circumstances, life happens. If you have to miss it, you have to miss it. But it's a lot of pressure, I think, to write a 68% final. I have also posted a grade estimator on there. It's an Excel spreadsheet with all the formulas already in there. If you want to know what grade you have to get in order to finish off with a certain mark, then all you have to do is punch in your values and it'll output your grade by calculating it both ways. So it may be a grade you think you are aiming for, it may be a grade that you already got, etc. So this will be more pertinent at the end of the term, but you could still plug in numbers and go, oh, okay, what do I have to get on this, 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 and this in order for me to get this? This individual got all these grades. They failed the midterm, got a 60 on the quizzes, bumped up to 70, and still finished with a 70. Okay, still finished with a 70, and this alternate grading did make a difference for them. 66 versus 70 is a big difference on your transcript. The last thing I want to say is, please, you know this is written down on the midterm exam tab. Please do not cheat. Some things in life are worth taking a risk. This is not. It's not worth taking a risk. You can recover from a low grade, guys. You cannot recover ever from an academic misconduct record on your transcript. You won't recover from it. So please, it's not worth it. I'm trying to protect you. I'm not trying to scare you. I'm trying to protect you. Okay, so let's start lecture. If you look at the first slide, we talked about last class, Mendel's P's was dominance. The red and white flower petal colors and the blood type was codominance. And the pink flower was incomplete dominance, which led to an intermediate expression. So dominance, codominance, and incomplete dominance. We'll cover those. Now, we also talked about what is the difference between dominant recessive? What does big R, little r mean? What do these different alleles mean? What they mean is differences in sequence. differences in the sequence. The sequence difference can be a point difference. It can be deletion, insertion, inversion, anything. Any difference in the sequence of the gene will lead to a different allele. So what is the impact of this mutation? What does this mutation mean? What does it have? Well, here's what can happen. This is no mutation. This is silent, nonsense, and missense. This is the normal DNA sequence, which codes for a codon that makes a specific amino acid. So in this case, it's lysine. A silent mutation means that when the DNA sequence changes, this is a point mutation, GC changed to AT, then you get the production of a codon, but codons are redundant, right? There's redundancy in the codon. So this codon codes for the same amino acid, so it doesn't matter. It's not going to make a difference in the protein. Nonsense means that the mutation is such that it creates a stop codon. A stop codon is something that says, stop translation, don't make any more protein. So this is premature termination of translation. Translation stops prematurely, and so you get the production of a shorter protein that may or may not be functional, depending on where the stop is. But for the most part, this type of nonsense mutation leads to a non-functional protein. It just depends on where the mutation is. If the mutation is far, down the road, then maybe most of the protein's already made and it's still functional. You get good luck. Missense is when the amino acid actually changes. Don't worry about conservative and non-conservative, just pretend it's not there. That's too much detail. Lysine changed to either arginine or threonine. This can have an effect, but this also depends on what the role of these amino acids are. Are they playing an important role in this domain that's binding to a DNA, that's this driving transcription, that's driving synthesis? For example, is the mutation in the 3' exonuclease domain? Is it in the 5' synthesis domain? Is it in the binding domain of polymerase? It just depends. You have to ask the question. You're in university, you're going to say, okay, there's a mutation, so tell me more. I need more information to tell you what the effect of this mutation is, right? This is what you need to know. So we were talking about mutations, point mutation. And generally, the word mutation is reserved for diseases. There's a mutation, therefore, this causes a disease. What if it's just a difference in alleles that leads to a different eye color? Would you say that people with blue eyes have a mutation, or people with this have a mutation, brown eyes have a mutation? No, you don't. They're different versions, right? But ultimately, there's still changes in sequences. So some people refer to these changes as SNPs, or many do. So SNPs are single nucleotide polymorphisms. Poly means many, morphism means different types. So you have differences in SNPs amongst us. So all of you will have a different pattern of SNPs, and I will have a different pattern of SNPs because we're individuals. So our pattern is different, but what remains similar are the patterns shared by your ancestors. So these companies that look for ancestry, they're looking at SNPs in your genome. Single nucleotide difference in DNA sequence, most common type of genetic variation among people, but it is quite consistent in your family tree. It occurs normally throughout the genome, and it occurs mainly between genes, but many SNPs can be observed in the genes themselves. It can have no effect, or it can affect your health. It just depends on the SNP. Most of them have no effect on health and development. However, some SNPs are associated with health and disease. And so in what way? Well, they can predict an individual response to certain drugs. So it's interesting. You can actually look at your SNPs and say, you will react better to these drugs than these. And that can be used to predict, instead of just shoving you with a whole bunch of drugs and, okay, come back next month, come back in a year, let's see if that worked. No, you know what? Most probable that these drugs will work better for you. You can predict susceptibility to environmental factors such as toxins. Some people, they're more susceptible to certain types of environmental factors as opposed to others. You can make that prediction. You can predict risk of developing particular diseases. If you send your genome sequencing, they'll come back with these numbers for you. You have this SNP pattern, you have a 25% chance of getting bladder cancer. Like, they'll literally say that in the report, which is scary, but what do you do with that information? This is why I would never get my genome sequenced, ever. I don't want to know. Because first of all, I don't even know if that's that accurate, and I don't want to be stressed. I want to live life not knowing. So, but you can do that if you want. So we talked about SNPs, we talked about mutations, the effect it can have on the amino acids. So ultimately, let's look at an example. Let's put all this together and look at an example of how different alleles can affect a phenotype. And we're going to look at eye color as an example. Eye color is very difficult to explain, actually, to be honest. I've tried to simplify this as much as I could, and you know the bulk of it. This is going to be the bulk of it right here. So eye color trait is polygenic. The trait is polygenic because it means that there are many genes involved in this phenotype. Poly means many. There are about 15 different genes. But the two key players are OCA2 and HERC2, the two key players. Okay, that's the first thing to know. Second thing to know is eye color involves two processes. One is production of melanin, how much melanin is being produced in the iris. And the second thing is how is the light being scattered as it's coming in. So the proteins in the eye will absorb certain wavelengths of visible light and they'll reflect back. So depending on what version you have of those 15 different alleles, you will reflect light in a different way. So those are the two main factors that lead to eye color. We're going to focus on melanin production because the rest of it you can just pick up an article and read it. So OCA2, this is allele type 1 and allele type 2. If you have allele type 1, then this is a gene that is transcribed and translated into a protein, and that protein is called a P protein. So OCA2 gene codes for P protein. The P protein, what it does is it is involved in the maturation and the maintenance of pH of melanosomes. Melanosomes are organelles in your melanocytes. Melanocytes are the cell, and melanosome is the organelle inside that cell, and it produces melanin, which is what gives the iris a dark color, the dark pigment. So melanin is a dark pigment, and that's what it gives it the color. If you have OCA2 allele type 2, then you have a different sequence, as you can see, and this allele produces less protein, less of the P protein. Less P protein means that you get less maturation of the melanosomes and less production of melanin. That's all it is. People with blue eyes, green eyes are not making blue and green pigment. You're either making a certain degree of melanin or you're not making a certain degree of melanin. So you're either making a lot or you're not making any. Those are the extremes. And then everything in between, right? It's a distribution, very similar to hair color that you saw, right? It's a distribution. So it's not like everyone just has brown, green, there's a range. So it depends on what levels of melanin you're producing. You're either producing melanin at a certain level or you're not producing melanin. There's no other pigment involved in eye color. But guess what the other key player is for eye color, and a big player? HERC2. HERC2 is the gene that regulates OCA2. It regulates OCA2. And how does it do that? It does it in an interesting fashion. This is HERC2 enhancer region. This enhancer region enhances or it activates the transcription of OCA2 gene by interacting with its promoter. It interacts with the promoter. This is the promoter of the OCA2 gene. So the mechanism is like this. Transcription factors come and they bind to the enhancer of HERC2. This binding will activate the promoter. And another event that takes place for activation is the following. Did you guys know that different regions of the DNA can form loops and different shapes and go and interact with other regions of the DNA and activate them? That's a cool mechanism of activation. So this enhancer region can form a loop, and this loop directly interacts with the promoter of OCA2. And now it says, go, make and transcribe this gene to make P protein and produce melanin. That's what HERC2 is saying. So based on this mechanism that you've learned, what type of SNP do you think people with blue eyes have? Do you think people with blue eyes have a mutation or a SNP in HER2? They do. They do. Because when there's a mutation or a SNP here, then this can no longer work, which means that it cannot activate OCA2. So P proteins are not being produced. So even if you have a functional OCA2 gene over here, one that will produce a lot of P protein, if your HER2 locus is mutated, you still have blue eyes. So you're like, but you have OCA2, you can make it. You still have blue eyes. So people homozygous for this will have blue eyes. Or even heterozygous. So again, look at the range, right? If you're heterozygous for HER2, then more tendency to have eyes towards the green color. If you're homozygous, blue color. If you're homozygous normal, then darker color. But that will depend on what, guys? It'll also depend on this allele. What about this? HER2 is like, I'm active. I can turn this thing on, but what is turning on is allele 2, which is not making P protein anyway. Do you see the combinations? But you've learned the foundation. You learned the foundation on how to understand eye color. This was the absolute bulk of it. So now let's move on to incomplete dominance. That leads to an intermediate expression. And what that means is the following. You have allele A and another allele, A and B, or it can be denoted as A, big A, small a, but that's reserved for dominance. Some scientists like to use the same letter but a different subscript. I'm using different letters, A and B, A allele and B allele. But they code for a similar protein, the same protein, it's just a different version. So A, if you're homozygous for A, then you make this protein, which is an enzyme involved in the biosynthetic pathway to make the red pigment. Pigments are not coded in the genome. You need to go through a pathway, a signal transduction pathway, to make the red pigment. So this is the protein involved in making red. If you're heterozygous for A, like you have one copy of A, one and B, then you make less of the protein because only one allele is making it, not the other. So you have less protein that is involved in the production of the red pigment, which is why the flower appears pink, because you're making less of the red. This protein is on jumping to another pathway that's making pink. It's just making the same pathway, but just less of it. If you're homozygous for allele B, well, you don't have any of this protein production, so no enzyme to drive that production of the red pigment, so the flower petals are white, no red pigment. So red, pink, white. A great example of incomplete dominance in humans is that of Tay-Sachs disease. So we actually have diseases in our body that follow the same pattern of inheritance, the way that flower petal color did. And that's Tay-Sachs. Tay-Sachs is a disorder of the central nervous system. And if you have severe Tay-Sachs, you can have paralysis, blindness, and even death. And the way it happens is through these accumulation of fatty structures. Your neurons are making these fatty structures that they require for their membrane, they require for the normal function of the neurons, and these fatty structures play an important role. However, when they start to accumulate, then they start to cause damage. The synaptic connection, the neurons can't communicate, and therefore you get these symptoms of paralysis and blindness and death. So to understand how this is happening, a couple of things you need to understand. First is that all molecules in your cells have a turnaround time. So in other words, they turn around. They're synthesized, they function, degraded, and then made new again. Synthesize again, function. It's just like recycling in your house, right? When you recycle at home. You use up your stuff, you send it for recycling, comes back new. Same thing. It is recycled. That's a normal process. So who does this recycling? Well, a bunch of systems come together, but before recycling, these fatty structures have been broken down into their individual pieces, individual parts, i.e. the elements, carbon, nitrogen, oxygen, right? Sulfur, phosphorus, all that stuff. And the organelle that does this are known as lysosomes. Lysosomes. Lysosomes are organelles that digest these particles, i.e. chew them up or digest, and then they release all those elements and then that gets recycled. But what do they need? They need enzymes to do it. There are enzymes inside the lysosomes that do this. And one of the key enzymes in the brain that is involved in this turnover process is HEX-A. HEX-A is a enzyme that is involved in that. So when you're big R, well, you are making HEX-A, just fine. If you're big R, little r, you're making less of HEX-A because one of your alleles is not making HEX-A, it's not working. If you're little r, well, you're not making any HEX-A, so this fatty structure is now accumulating. So now you have severe Tay-Sachs. So you have mild here because you're making some HEX-A, no Tay-Sachs here. This is very similar to the flower petal color. And students on the exam, they're like, oh, well, I don't understand. You want me to compare Tay-Sachs with flower petal color? What do they have anything to do with each other? Incomplete dominance, intermediate expression. You could see this as the red flower, pink flower, and the white flower. But in this case, it's a disease in our brain. Codominant alleles are equally expressed in heterozygotes. So let's look at this. Here you have type A antigen on your red blood cell, type B antigen on your red blood cell, type AB and type O has no antigens. So the way you determine your blood type is the type of antigens that you have on your red blood cell. If you're type A, you actually have anti-B bodies in your plasma, so your blood is circulating with these antibodies. If you're type B, you have anti-A antibodies. If you're AB, you have no antibodies because you're not going to mount an immune response in your own red blood cells. If you are neither A or B, you don't have any cell surface antigens, but you have both the antibodies circulating. So here's, well, back in the day, about 100 years ago or even more, people were literally dying of blood transfusion because they didn't know. Blood is blood. Blood is not blood. The type is important. So what was happening is they were being transfused by the wrong blood. The antibodies that were circulating in that individual would mount an immune response. That's what immune system does. It thinks it's foreign. It's just doing its job. It mounts an immune response, and then guess what? The red blood cells lyse. They burst. It's called hemolysis, and that's how you die. Your red blood cells are literally bursting. AB is the universal recipient. O is the universal donor. O minus is the ultimate, right, universal donor, but we're not going to talk about the rhesus factor, positive and negative. Just know we're going to focus on the antigens, and we're going to focus on the AB antigens, and we're going to focus on what allele is responsible for delivering these antigens onto the surface. That is what determines your blood type. So the glycoproteins and the alveolar blood type. Here you have a red blood cell membrane. This purple is a protein, and this part here is the sugar group, or glyco. So this is glycoprotein. Your cell surface markers are actually glycoproteins. But your antigen, your actual antigen, is only the last terminal sugar. The antigen that gives you A, B, or O is your last terminal sugar. And so look at this. This is all common to all of us. We all have all of that in common. What's different amongst us are these last terminal sugar. And that determines your blood type. So who is adding that last terminal sugar? That is the enzyme that's coded in the DNA. That is actually the differences in your alleles. Not the actual A, not the actual B, not the, those are not the actual allele that is different in sequence in your chromosome is the gene for glycosyltransferase. Glycosyltransferase is the enzyme that adds the last terminal sugar. And it has differences in sequence. If its sequence is that of type A, it has a specific sequence, then it will go and pick up N-acetylgalactosamine and put it on to this structure here. If the sequence is type B, it'll pick up galactose and add it here. Now this individual has B antigens. Type O, guess what? Type O does not add any antigens. So what does that tell you about type O? You've learned enough to predict. If you say, oh, type O, there is nothing there. But that's the job of glycosyltransferase, it's supposed to do that. So I wonder if people with type O, if you were to make a conclusion and look at their glycosyltransferase allele, what do you think you could predict? Yes. That's a good one. They don't produce it at all. Yes, that's a really good one. They don't produce it at all. Or they produce a non-functional type, right? They have a mutation such that glycosyltransferase is not functional. And that's what it is. One base pair, the most common type of mutation is one base pair is out. It's gone. Boom, frame shift mutation. Now the codons cannot be read in order. And so you get a non-functional glycosyltransferase. But would you say that type O people have a mutation? No, right? You would say they have a different version. But some scientists will say there's a mutation in the glycosyltransferase that leads to this type O. And yeah, people like to switch it over to there's a SNP. It's a different version. They're not mutants. So yeah, that's the reasoning. Now let's finish off with sex-linked characteristics. So sex-linked means that the gene that is leading to the trait or the disorder or the disease is on the sex chromosome. And we're going to be focusing on the X chromosome. So X-linked recessive diseases or X-linked dominant diseases. So females have two X's, males have an X and a Y. I will again be using male, male, and female, not offending anyone. So do you guys think that X and Y are homologous? No, they're not homologous. But when we look at mitosis and meiosis and we're talking about homologous pair separating, homologous, we just clunk in the X and Y in there too, right? And said, yeah, they're homologous too, but they're not technically homologous. X is a much larger chromosome, Y is a much shorter chromosome. They have different gene expressions on them. It's different genes and they express different proteins. So they're not the same. Which is why sex-linked characteristics follow a different inheritance. It's because of the fact that X and Y are not homologous. So here you have an unaffected father, a carrier mother, and then from the cross, you can see what proportion of the progeny or the offspring are going to have the disorder, the trait, or the disease. So females can be carriers. So for an X-linked recessive disorder, the mutation has to be on both the X chromosomes in order for you to have that trait or that disorder. So that's what's called X-linked recessive. Both X's have to have it. So females can be carriers. If one has it, they don't have the disorder. Both have to have it. This is why males cannot be carriers because they only have one X. They don't have another X to compensate. So this is the X. If this is color blindness or hemophilia, this male will have that. Females can be carriers, males cannot be carriers. X-linked recessive disorders can have a high probability of appearing in the offspring. They have a high probability because of the dad. The dad does not have a functional X if his X is affected. Carrier female, affected male. So if you have both of these, and if you cross them, do the exact cross the way I taught you. Write these down, put them on the horizontal bar, on the vertical bar, put these in there, write them out. Now look at the genotypes you have, and then tell me what the phenotype will be. It's the exact same thing as that purple and white flower. Don't overcomplicate it. Just know that this is different in that this is X and XY that I'm going to put down. These are not autosomal chromosomes. So you put that down, and then you say unaffected female, unaffected male, affected male, and affected female and affected male. 50% chance that the offspring will express an affected phenotype. 50% chance. If you look at this, both of them have one of their chromosomes affected, right? And the other one not. So both of them have one affected, one not affected, one affected, one not affected. 50% chance. Now take this exact same thing and convert it to an autosomal chromosome. Autosomal are your chromosomes 1 through 22. Look at the chance. It reduces by 25%. The reason for that is because, look guys, carrier female, affected male. Unaffected female, unaffected male. In this case, she's also unaffected. So this should also say unaffected. I think that would have been a better comparison. Unaffected, affected, unaffected, but the male now is also unaffected because it's autosomal. It's not XY. The male has another chromosome that's normal. It's not the Y chromosome, right? So if you do the same monohybrid cross, you come up with this distribution, in this case, 25% chance. So X-linked recessive disorders can have a high probability of appearing in the offspring as opposed to autosomal. Red-green color blindness is an X-linked recessive disorder. Are we going to call it a disorder or a trait? What do you guys want to call color blindness today? Disorder, not a disease. Disorder or trait? How many say disorder? How many say trait? I say trait. Okay, so yeah, it doesn't matter, guys, honestly. Red-green color blindness prevalence. It's 1 in 12 in men, 1 in 200 in women. Why do you think that is? Why is there such a big difference? Meaning it's more prevalent in men. Yes. Exactly, perfectly said. Men, again, have only one X. So if they have the colorblind gene on the X, they are colorblind. But if the female has a colorblind gene, she may not be colorblind. So in this population, there are carriers. In this population, there are no carriers. So this is why men have a tendency or higher prevalence to see that. So X-linked recessive disorders, more prevalent in men than in women. All right, so it was a pleasure to teach all of you. I hope that you can come to me and ask me any questions you have before the midterm. Make sure that you guys know all the help sessions that are available to you up until October 18th. You have a bunch of help sessions. Look at the announcement and come to all of them. We're here to help you. All right, so on that note, I will see you guys in February. Thank you.